All right, welcome human biology class. This is the final attempt at some semblance of lab content. Your videographer today is my daughter Becky, and hi. There are three things we're going to do today. The first one has to do with the bone lab and or the musculoskeletal system. I had mentioned that bone is made of collagen and calcium phosphate. Collagen giving it the rigid part, calcium phosphate giving it the hard part of bone. Together they're a fairly tough, durable thing. So if I take this rib bone, these are rib bones, had ribs the other night. These are from a pig, okay? And so if I tap it here, you can see that's a fairly hard bone. So these are all from the same rack of ribs, and I took that same two more bones from that same rack, and I put them in 10% hydrochloric acid for overnight and a day, and basically turned them into this. Let's see if this one's a little rubberier. Okay, so what we did, taking this bone and soaking it in 10% hydrochloric acid, we dissolved away the calcium phosphate part of the bone and left just the collagen part, which is the flexible part. Okay, so it's the same bone, just missing that calcium phosphate part. And so that it does a good job, I think, of illustrating the difference between, well, I mean, I could break it, but it would splinter. Between the two parts that make up bone, calcium phosphate and collagen together, hard. Take away the calcium phosphate, just leave the collagen, and you get a very rubbery bone. It's also how you make rubber chickens. Just kidding. Okay, so the next thing we're going to do is dissection of a sheep brain. And this is a sheep brain, so it would be oriented in the sheep like this. This would be the back of one eye. This would be the back of another eye. And this would be the brain stem right here. And this would go down to the spinal column. Of course, sheep have a brain that's situated like this, and people, it's going to be a little bit more upright like this. But I wanted to just point out some external structures first. First, right here, this is the olfactory bulb. And this part right here would go right into the nasal cavity of the sheep. And you can see, I mentioned before that olfaction, your sense of smell, is the only sense that is connected directly to long-term memory. That's why smells can conjure up such a potent memory. So right there is one. I'll take this other sheep brain, see if it's got both of them intact. They're a little mangled on this one too. And there's one. It shows better here. So there's one right here, and there would be another one right here. Olfactory bulbs, eyeballs. The brain is covered by a really tough membrane called the dura mater. Collectively, there are three membranes which make up the meninges. An infection of the meninges is called meningitis. It can be either bacterial or viral. Interestingly enough, viral meningitis is not nearly so nasty. Bacterial meningitis is what kills people in a few hours. The outermost layer is called the dura mater, which is Dutch for tough mother. And it is a tough mother. I mean, that's this stuff right here. And I mean, it's tough. And underneath it would be cerebrospinal fluid. So like your heart, your brain is contained in a sac, fluid-filled sac, that protects it. The meninges descend down here, deeper in, and I'm going to see if I can peel them back. I might have Beck pause the video from time to time, not right now, but if I need to do some dissection that you don't need to watch the minutia of. But I'm going to see if I can get that out of there. And what I'm doing is I'm cutting that meninges. I'm trying to cut the meninges. The dura mater is a specific one I'm trying to cut. And I'm not succeeding, so I'm going to go to the scalpel here. It's always better to use a scissors when dissecting, because then you have less of a chance of cutting something you don't want to cut. Okay, I'm getting close. Like your finger. There we go. So I'm going to pull that meninges off that side. And on the brain, so we would have what's called the temporal lobe, the parietal lobe, occipital lobe, parietal lobe, occipital lobe, frontal lobe. It looks like just a bunch of chewed bubble gum, but there are ridges and valleys called gyri and sulci here, and they're in specific places. There's not a randomness here. And so brains are mapped according to where those ridges and valleys are in them. Some people are amazed that sheep even have a brain. If you've ever raised sheep, some people say they can drown just by looking up during a rainstorm. But they do, and here we have it. So I'm going to try to pull the dura mater off the other side, and it is tough, I'm telling you. Not easy to work with. Okay, so there we're getting it off. There's the dura mater off the other side. And there are lots of nerves and blood vessels that come through holes in the dura mater as well. You see me pulling some of them loose here. You can see some more nerves down here that are coming out of the brain directly. We talked about the brain, the 43 nerves, the cranial and spinal nerves. So some of the cranial nerves you can see right here. Here's a blood vessel, not here, but right here's a blood vessel that would go into the brain. So right here, this is another very fine layer on the brain here. I'm going to take that and see if I can just, I pulled it off now. So underneath the dura mater is a layer that looks like a spider web. It's called the arachnoid layer. And underneath that is a very delicate layer like cellophane right here, just on the surface of the brain. I think maybe you can just see that under the probe. Is that coming in, Beck?
You're a little bit up. Come down.
So down here?
Yeah.
Okay. Right there. Very thin, fragile, delicate membrane. It's called the pia mater, delicate mother. So dura mater, tough mother, arachnoid, and pia mater, or delicate mother, are the three membranes that make up the meninges that cover the brain. Okay. Again, olfactory bulb, and I'm going to pull some of this off just a little bit more. And some of it came off a little bit better than I wanted it to come off, but we'll see what we can do here. Why don't you pause it for just a sec, Beck? Okay, so folks, I took the other sheep brain, and here's one eye, here's the other eye. I've turned it over, so this is the brain turned over. And the reason I wanted to show you a particular structure, it actually got torn in the other one, so I wanted to show you here. Right here, I started to cut it right here. This portion coming out of this eye, right here, and this portion coming out of this eye right here, these are the optic nerves. And they form the optic chiasma right here. And if I take this, I don't know if I can get this apart or not, probably not, without tearing it even more. You can see that they cross over, so that normally what you do with your right side is controlled by the left side of your brain and vice versa. But the eyes are a little different. What you see with your, well, it'd be the right eye here, this would be the sheep's right eye. What the sheep sees, or what you see with your right eye, goes to both sides of the brain. What you see with your left eye goes to both sides of the brain. Excuse me. In your lab manual, under the senses lab, there's a little piece there that has you look at your blind spot. And that blind spot is made possible because of the fact that what you see with each eye goes to both sides of the brain. Okay, the blind spot is actually there because of a spot in the back of your eyeball, which we'll get to in a second. But what you see, the field of vision that you see, is there because of the optic chiasma. Okay, so that's that little X right there, the crossover. Okay, okay. So what I've done here now, let's see if I've got most everything off here that I wanted to get off. So what I've done here now is I've cut the brain in half, and I'm going to continue that cut, and we'll look at some specific features here in cross section. Yeah, it actually came out pretty good. Okay. So this structure right here, this is the cerebrum. This structure right here that kind of looks like cauliflower, this is the cerebellum right here, this structure right here. Is that okay, Beck, if I lift it up this high, or is that out of focus?
It's good.
Okay. So this structure is the cerebellum. The cerebellum is responsible for mainly for coordination. Those of you, if you've seen somebody that can walk a tightrope or something like that, the Wallendas or something, this is a part of the body that they've developed really well to allow them to be able to do that. The cerebrum in humans, this is 4/5 of the brain, the cerebrum right here. This has gray matter around the outer 1/4 of an inch of it, and the rest is white matter. Remember the spinal cord, the gray matter was on the inside, the white matter on the outside. There's a crossover here. You've got gray matter on the outside and white matter on the inside. Of course, this is a sheep brain. It has no gray matter, or very little. At any rate, this structure right here, this is the corpus callosum. This is the crossover communication bridge between the two hemispheres. Okay, so right here, this allows for communication between the right and left sides of the brain. Many things that you do require sensation and direction and processing from both sides of the brain. I have a friend whose daughter had severe epilepsy, and so they cut her corpus callosum. They had tried everything else, and nothing worked. So they cut her corpus callosum, and in an attempt to isolate the epileptic seizures on one side of her brain, it ended up not working. But one of the results, until she sort of reprogrammed her brain a bit, was that she would walk up to a door and know she wanted to go through that door to get to the other side for the bathroom or the kitchen or whatever. But she couldn't remember how to do it. And so the desire to get through the door would be on one side of the brain, the knowledge of how to manipulate the doorknob on the other side of the brain, and the two did not communicate. So that's what the corpus callosum can do for you. Okay. I'm just looking at both sides of this brain to see if one side, this side might show up just a little bit better. The next structure that I want to show you is the thalamus. If you can kind of see this rounded structure right here, that's the thalamus, and there's actually an opening here. I'm just touching it a little opening right here. And the floor down below that rounded structure is called the hypothalamus. So the thalamus sort of decides what to send up to the higher brain function and where to send it. The hypothalamus controls your body's internal environment, if you will. It controls thirst, hunger, body temperature, things like that. So real important functions right here. If I go back to this other side of this sheep brain, right here we see the pineal gland. As people ask what that is, and pituitary gland here. Not going to talk about those too much. Right here I wanted to point this out though. This is the corpus callosum, and I'm going to sort of pull the two parts apart just a bit, and you can see right there there's a space. Is that showing up good, Ben? This is one of the ventricles of the brain, and this is, among other things, where cerebrospinal fluid is produced. Okay, there's one on one side. On the other side, we would find the other one right in there. Okay, so that is the first and second ventricles, or the lateral ventricles. Cerebrospinal fluid is produced there. It is produced here in the third ventricle. This is a little smaller, a little harder to see. It is produced here in the fourth ventricle, which is very, very vague, but it's right here. And that cerebrospinal fluid then flows down the spinal cord, flows back up the spinal cord, and is dumped into your blood circulatory system in the arachnoid layer. So there's a circulation of cerebrospinal fluid. It's constantly kept fresh. People that have difficulties with the draining of that cerebrospinal fluid will have it build up on the brain. It's called hydrocephaly. And whenever you have either cerebrospinal fluid or blood building up on a part of the brain, it puts pressure on it, and it can kill off the part of the brain where pressure is being applied. So it's very important that the cerebrospinal fluid be produced, because it protects the brain, flow down the spinal cord, back up, and get recirculated into the circulatory system. So then we have a bulge right here. This is the pons, the medulla oblongata, and the spinal cord. And these structures sort of are, I was going to say gateways, not gateways, but switching tracks, switching centers, which decide where certain stimuli are sent, what to do with them, that kind of thing. Okay. All right. What else did I want to talk about with the brain? I want to pause it there for a sec, Beck. Okay, folks, I remember one last thing I wanted to mention. Right here, the medulla oblongata, which connects the brain to the spinal cord, this is where centers for breathing and heartbeat are maintained. And so a short distance down the spinal cord then, those nerves that control those things exit the spinal cord and go to your diaphragm, to your rib cage, to your heart. That's why people will often talk about wanting to make sure that your spinal column stays intact above C5 to stay alive. Cervical vertebrae number 5 and above is where those nerves come out. If your neck is broken at those spots, you will die because your heart will stop beating, you will stop breathing. Okay, and I think that is about all I wanted to say about the brain. I'm just going to slice this one in half again, real good, just to see. It's to see what see how I do. I'll put it in the middle here, and sometimes some of these structures show up better because one cuts it just a hair differently one time compared to the next. So let's just give this a go here and see how we do. My tongue is hanging out because that shows I'm really sincere. She just punched me, my own daughter. Yeah, not bad. Shows that lateral ventricle, first or second ventricle, really well. The thalamus, here's shows up the third ventricle just a little bit better right down here. So the thalamus, the third ventricle, the pineal gland, not quite as good. Pons and medulla oblongata, here's the cerebellum, shows up pretty good. And of course the cerebrum here, corpus callosum. And yeah, how about this side? Yeah, pretty much the same. Yep. Thalamus, yep. All right, why don't you pause it, Beck? We'll get the... All right, folks, the last dissection we're going to do is the cow eye. Remember now, I've got my eye on you. Huh?
Can't see that.
Can't see that?
Nope.
How about
Yeah,
that?
that's good.
Got my eye on you. Okay, so this is a cow eye, calf eye, and this has been taken right out of the skull. There's a lot of connective tissue here, and I've cut some of that away. And here we can see some of what are called the extrinsic muscles. These are muscles that would allow the eyeball to move around in the socket. And some of them are going to be attached up above and below for the eyelids and lashes and things of that sort. If we turn the eyeball over right here, this structure, rounded structure right here, it's kind of tough. This is the optic nerve coming out of the back of the eyeball. Okay, and so we're going to look at the eyeball. So this would be the cornea. This would be the sclera. So this would be the white of the eye. This would be the cornea. The cornea has many layers to it. Some of the outer layers are called the conjunctiva. And bacterial infection of the conjunctiva would be called conjunctivitis, or more commonly known as pink eye. When you have LASIK surgery, it is this outer portion of the cornea that will be cut. If you are nearsighted, your eyeball is a little bit too long this way, and so you focus an image in the wrong spot. You're actually trying to focus it beyond the back of the eye. So what they'll do is they'll shave some off of the cornea here to reduce that distance and return normal vision to you. If you are farsighted, it means you can focus on things farsighted. You can focus on things far away, but you can't focus on things up close. Your eyeball is too short. And so in that case, they'll take a little slice off around the perimeter so that the eyeball actually bulges out a little bit and restores normal vision. Okay, so nearsighted, you can focus on things up close but not far away. Farsighted, you can focus on things far away but not up close. When you get to be, well, at my age, 20 years ago, well, I'm going to talk about what happens to your eyeball when you get to that stage. So what I'm going to do here now is I'm just going to cut back on some of these layers just a little bit more, try not to hack my hand off here. And what I want to do is cut into the eyeball, and we want to look at some of the layers, because really the way to think of an eyeball is just to think of it as two layers, as having two layers. So there I've removed some of that. If you like Andrew Zimmer's Strange Foods or whatever his program is, this is the stuff that he would eat. So what I'm going to do is about a 1/4 inch away from or into the sclera, I'm going to cut maybe 1/8 of an inch. I'm going to cut in, and it's going to pop. There's going to be some goosh of fluid as I make that cut. That's because there's fluid in the eyeball, and the amount of fluid in the eyeball helps dictate its shape. Actually, I think I'm going to take the smaller scissors here. And here we go. So I'm trying to cut around the perimeter of the eyeball there. So did you catch that squishy, gurgly sound? You just can't fully appreciate all that's going on here unless you can feel it. All right, there we go. So what I've done is I've just taken the front of the eye off, and let's turn that over, and I want to look at it here because there's some things that are important to note. Now, Beck, I'm not sure how well that people can see this, but this hole here would be the, let me get this stuff out of here. So this hole right here would be the pupil of the eye. The iris would be this, the colored portion of the eye would be right here. Of course, on cows, they're black. They're very dark colored. Okay, and if we could look at this microscopically, we would see circular muscle making up this black portion we call the iris. If you think about what happens when you come from a dark room outside, that pupil shrinks inside because you're all of a sudden bombarding the back of your eyeball with all that light, and it doesn't like that much light, so it closes down the side of the size of the circle, the opening here, the pupil, to let less light in. That's achieved by these circular muscles that make up the iris. Sitting right, see if I can pop this rascal out. Okay, right here, I'm just going to take this out. This has been clouded by the preservative here that we use to preserve this. What I've just taken out is the lens of the eye. And this lens of the eye would sit, actually would sit like that, right there in that nice rounded little depression. Okay, right like that. And I'll take it out of the way. And I don't know, Beck, you'll have to say, can you guys see these, there's some lines here on this outside
Maybe a little.
part? Maybe a little. How about here? How about down here? Can you see them if I do that?
Yeah.
Okay. Those are radial muscles. So in the iris, there are circular muscles, but out here in what's called the ciliary body are radial muscles, and they actually attach right here around the edge of the lens. Okay, so the lens would sit in there. Those muscles would attach to the lens, and they're going to tug and pull on the lens. They're going to work when you go from looking at something half a mile away to try to look at something up close. Your ability to focus on that thing distant and then to focus on something up close and everything in between is dependent on those muscles tugging and relaxing on the lens to change its shape, all for the purpose of focusing whatever is coming through that lens on the back of the eyeball, which is where your sensory organs are. Okay, now one thing I got to point out is this lens should be crystal clear as this baggie in your eye. But the preservative has clouded it. This would be analogous to what a cataract would look like. And a cataract is a clouded lens. It doesn't let all the light or all the colors in. The person has to have more light to be able to see. Cataract surgery often involves just removal of the lens. You can actually see without a lens. You have to have corrective glasses, of course. I've heard now that they can put an artificial lens in that will even correct your ability to read near vision. So I'm not sure, but I've heard that. I haven't verified it yet. So that's what a cataract is a cloudy lens, and cataract surgery removes that lens and can put a replacement in, an artificial one sometimes, to take its place. Okay, so that's the front of the eyeball, and this cavity up here in front of the iris, but behind the cornea, would be filled with the stuff that you see in the bottom of the tray here called aqueous humor. And the amount of that that's in there would help dictate the size and shape of the eye. Okay? So the front chamber, there's two chambers to the eye. The front chamber would be filled with aqueous humor, and it would be plugged by that lens. Okay? So if we take that front part away and we look at the back part, now there's a back chamber here, and this back chamber is filled with a thick viscous fluid. I'll put some in my hand right here, called vitreous humor. Got something else in there too, piece of the back of the lens. So again, this is obscured by the preservative. It should be a clear jelly-like substance. You can see that it's kind of jelly-like, but that is called vitreous humor. And the amount of it that's here in the back of the eye would also help dictate the shape of the eye. Let's see here. I need it. Forceps. There we go. When you think of the eye, think of three layers. This back layer right here, that's all folded together now, this is called, what's the inside layer? This is where the rods and cones are, and connected to the optic nerve. Let's see if I can show you where it's connected here. I think it tore away a bit here. Don't you? Okay, right there. I'm going to get this piece that's torn away out of there. So this is some retina that I'm taking out of there. This would have rods and cones in it. But right here, where it's attached, okay, this is the retina. That's where the optic nerve would leave, okay? And if we flip this over then, that's also exactly where the optic nerve exits the back of the eyeball. Okay, when you focus an image on the part of the retina right here where it forms the optic nerve, there are no rods and cones there. That is your blind spot. So in the lab manual, when it asks you to look at the dot and the X, you are attempting to focus that image on this part of the eye, and that would be the retina. So, that would be the optic nerve, I'm sorry. So the retina is right here, and it's full of rods and cones attached to the optic nerve. Outside of that will be the ciliary body, which would go around and become this black stuff right here. It's called the choroid coat back here. And the outermost layer would be called the ciliary body, and that would go around, the sclera, and go around and become the white of the eye, the sclera of the eye. So it got the retina, the choroid coat, which becomes the ciliary body, and the sclera, which is the white of the eye. There are your three layers. Retina, I'll pull it right off now. Retina, choroid, maybe we can see it even a little bit better there. There's a retina. The choroid is the middlemost, and the sclera is the outermost that forms the white of the eye. In cattle and many animals, they have an additional layer called the tapetum lucidum, and I can separate that out. It's what makes that look pearly luster look in there. Can you see that back? Okay, so right here, if I lift it up, you can see I can lift that layer right up. Humans do not have this. We do not have eyes that glow in light. If you've seen deer or cattle or dogs, their eyes glow in the dark, and that's because light is reflecting off this tapetum lucidum layer that we don't have. Its purpose is to shine light back on the retina so they can see better in low light. The retina would sit right on top of that layer, and again, that's what perceives images. So, that's the main parts of the eyeball. Let me just take a quick peek back. Would you flip the page there? I just want to make sure I covered everything. All right, looks good, looks good. Yeah, there's another spot here that cows don't have because they don't read, that humans do. And if you ever think about it, if you pick up something to read it, where do you hold it? You always hold it right in front of your face. Try to read it, hold it the same distance away from your eye, but move it off to the side. you can't read the print. You can see it, but you can't focus on it. What you are doing when you hold that thing that you are reading right in front of your face is you are focusing that image on an area very near the optic nerve, but off to the side of it, in the back of the eye, somewhere like here, called the fovea centralis. It's actually in the center of something called the macula. And the fovea centralis is a spot where you have a particularly dense cluster of excuse me, of cones. And you have so many of them there that allows you to see things really, really up close. And so that's what you're doing when you're holding something where you want to read it. You are focusing that image there. You may have heard of somebody that has macular degeneration. That's when that area of their eye begins to degenerate, or retinal detachment. That's when the retina pulls away from the eye and it loses blood supply in an area. And oftentimes they'll go in with laser surgery and sort of burn an area around the retina to seal it back on and isolate that area that's been deprived of nutrition. It's probably dead anyway. But that's what those surgeries involve. The retina here has to maintain a good blood supply. And if it becomes detached or separated from the excuse me, the choroid, it's going to lose its blood supply in that area of detachment and die. Let's see. So we talked about LASIK surgery. We talked about cataracts, macular degeneration, retinal detachment. Floaters in the eye can be little spots of particularly thick areas in the vitreous humor, this thick jelly-like stuff back here. And they can slowly sort of fall through the vitreous humor and give you that look. If you've ever been hit hard in the eye and seen stars, that's actually sometimes you're looking at blood cells that have come out of a hemorrhaged vessel on the retina, and there's actually bleeding there, so obviously that's not a good thing. Can you think of any other questions people might have on eyes?
What about when you're having like a migraine and something appears in your vision?
Oh yeah, there can be all sorts of visual distortions with migraines, and typically that's not to do with the eye. That's typically to do with the processing of eye information in the brain. Okay, so that's not typically a problem in the eye. It's typically a problem down the way as this optic nerve contacts a part of the brain that's being affected by the migraine to give you the impression that you can see those visual distortions.
Fascinating.
Anything else? Think of? No? All right, folks. That's all she wrote. Thank you very much.